Hello and welcome back. It's Professor Hendricks, and today I'd like to talk to you about reading in a Gtf file and, more to the point, extracting a transcript sequence from a Gtf file and a genomic Fasta file. So what I mean by that is we're going to read in a genomic Fasta file and we're going to read in a Gtf file, and we're going to extract the transcript sequence by concatenating the exons for that transcript. But before I begin, I want to explain a couple of things. First off, Gtf files. So a Gtf file looks kind of like this. We have 9 columns, and they're the sequence name, which is going to be like the chromosome name. We're going to have a source, or the database from which the record came from. Feature. So feature can be mRNA, exon, 5' UTR. It has a lot of different features that you might describe features on a genome, but also features on a transcript that are transcribed from that genome. The start and end are one-based coordinates, and they're also one-based inclusive coordinates, meaning you start counting the first base pair of the chromosome as 1, and if you're talking about a substring, it's probably in the way that you would normally think about substrings. It's going to go from this position to this position, and it's going to include both those two positions that I just stated. It's going to be the more intuitive notion of substrings. There's also a score, which can have various meanings depending upon the type of data that you're looking at. I'm not going to go into score today, but for example, if this was a BLAST result, you might have a BLAST score, or if these were predicted transcript models, you might have some sort of score indicating the level of confidence in that transcript model prediction. The strand refers to the strand on the genome from which the record resides, or from which it's transcribed in the case of a transcript model. Frame refers to the reading frame. So this is only going to be valid for CDS records. CDS meaning coding DNA sequences. These are open reading frames that are actually translated and have a defined frame. And in case you're wondering, exons are definitely not always split at the precise codon boundaries. Exons are split anywhere they want, including in the middle of a codon. And so the reading frame can vary from exon to exon. And then lastly is the attribute column. The attribute column stores a lot of other information like the IDs. And so we're going to want to be able to parse information from the attribute column today as well. And the other thing I want to point out is transcript models. So here I am on Drosophila database called flybase.org. And if I were to type here in this upper right-hand column, I can type in this CNC and go into search. It takes me to this page that I'm at right now. Scroll down and I get a graphical representation of the gene. It's still loading here, but here it is, CNC. And I'm going to click gbrowse. Gbrowse is a sort of like a genomic web browser. It allows me to navigate through the genome. And I can see here that this CNC gene, which by the way is the ortholog or homolog of NRF2, it's involved in oxidative stress, and it's a pretty important transcription factor. You can see that there are lots of different isoforms, and if I want to grab a particular one of these, I have to be very careful which one I'm referring to, and the actual encoded protein is also going to vary depending upon which one of these isoforms I extract. And these last 4 certainly look very similar. Maybe there's a subtle difference between them that I can't really tell. For example, right here, this one looks like it has a longer exon. You can see with my mouse right here. That looks a little longer, and maybe there's other slight variants among these, some of which do affect coding sequence, some of which don't. So I'm going to try to get this RC. So RC is the first, I believe, identified long isoform of CNC, or it's one of them, and I'm going to try to extract that and concatenate these exons. And the other part of that is I want to reverse complement. So you notice this gene is in the reverse complement. So really what I want to do is I want to concatenate these exons, but I actually want to concatenate them in reverse order. So that's going to be a little tricky. How we would do that, there's a couple ways we can do that. One way is to just go ahead and concatenate the exons right away in the order of their genomic position, and then simply reverse complement the result. That's probably the easiest way to do it. So let's just do that. So let's get started. I went ahead and copied together some of the code that I've done in the past videos. So if you haven't seen my reverse complement video or my reading large Fasta file video, I have all of that here. So I have the read large Fasta, and I have a bed file, read bed file as a placeholder. I'm going to modify that to read in a Gtf file. But I also have reverse complement, complement reverse, and these are all different ways that I'm able to parse the exons, reverse complement them as I read them. And first thing I might want to do is I might want to start off by thinking about what do I want my input files to be, and what do I want my usage statement to be. And again, this is just a placeholder. This is actually from the Fasta file, but I do definitely want to have a Fasta file. I want to have a genome Fasta file. So I'm going to put genome Fasta file here, and I also want to have a Gtf file. And I also want to have a transcript symbol, let's just say. Meaning each of these transcripts have a symbol. So in that particular example that I was just showing you, this CNC example, the symbol would be CNC dash capital R capital C. And if we go back to this flybase GTF file that I've downloaded, I'm going to turn off word wrap there, and we can start to see what this file looks like. We have the seq name or chromosome name at the first column, source, sequence type, start, stop, score, strand, and frame. Again, this frame is a dot in this case because we're not looking at a CDS feature here. And then we have this attribute column which has all this extra information. So it has gene ID, gene symbol, transcript ID, transcript symbol. I mentioned CNC, so I'm going to search for that within less. That's just typing in a /, and then I can type in CNC-RC just like that, and see if we can find this particular record. There it is, and we can scroll over to the right and see that it contains 5' UTRs, exons, and you notice that the 5' UTR is annotated and has a particular start and end for that, but it also has a corresponding exon record for that 5' UTR. So it does keep track of which exons and what part of exons are UTRs versus coding sequence. But for our purposes of extracting the transcript, it would suffice to pay attention only to these exon records as we go through. And so let's keep that in mind as we go. And essentially what we want to do is grab something that matches this transcript symbol. And I'm going to go ahead and copy this because this is important. We're going to have to use this format here in order to properly extract that from each of these columns as we go in such a way that we will hopefully get the right transcript ID and concatenate the right exons. So let me go back to my code. So with this usage statement in mind, there are expected to be 4 terms in my command line argument. In other words, the 0th term is going to be the script name itself. The term indexed by 1 will be the Fasta file, the term indexed by 2 will be the Gtf file, and the transcript symbol will be the term indexed by 3. So a total of 4 terms, 0, 1, 2, 3, 4. So I'm going to require this to have 4 terms, and then I want to specify these. So instead of file name, I'll be more specific, and I'll say genome Fasta file. And I'll also put Gtf file as sys.argv of 2, and the transcript symbol sys.argv of 3. Now I believe this should be good, and the next thing I want to do is I want to read in my genome Fasta file. So I want to basically put this here, and I'm going to just go ahead and call it genome instead of sequences. I'm going to be more specific, and I want to change this to genome Fasta file. And I want to go ahead and clear this stuff up here. So really what I want to do is I want to have some sort of way of retrieving the exon coordinates for any gene and concatenating them. And there's a couple of different ways that we could do this. We could either pass in the genome itself into the GTF script and extract our exon sequences as we go. And that might be the best way to do it. I'm willing to do it that way. Seems reasonable. Another way to do it is to have our Gtf file parsed in such a way that we retrieve the transcript models. And you know what? Let's do that way. So I'm gonna have some data structure called transcript models, and that's going to be read Gtf file. And I'm going to do it like that. Maybe if I want to stick to camel case, this is one of these cases where I'm debating whether to stick to camel case or to actually, I'll just stick to camel case. Well, you know what? I guess here I have this underscore, so I'm going to go back to underscore. Oh boy. The more important thing is that we're consistent, and I'm clearly not being consistent, but maybe I can fix that. Okay, so clearly I'm going camel case with my variable names and underscores with my function names. I'm just going to roll with that for now because I don't want to change it. So when I read my Gtf file, I want to modify, I'm going to just replace, find and replace, again escape %, and everywhere I have bed, everywhere I have bed, I want to replace with Gtf. Now out of the box this isn't going to work because we're expecting a completely different format, but I want to change it. So what I want to do is I want to replace this with the columns of the Gtf file, and just to recall, these are what those columns are. And it's going to be Chrome source, and I'm just going to call it Chrome. Technically it's seek name, but you know what? I'm just going to call it Chrome. And this is going to be source. I'm just going to put type is kind of a keyword in Python, so I'm going to call it seek type. And then I'm going to have start, end, score, strand, and then frame, and then attribute. Maybe I'll just call it ATTR for short. And it's going to be splitting on tabs. And what I would like to do is do something different. I'm going to store something a little bit different than what I did before. And what I want to do is I want to first check if the seek type is an exon. If it is an exon, then I want to work with it. So as I mentioned before, if my goal is to simply extract the transcript sequences, all I really care about are these exon features. The other features, though useful in their own right and in their own appropriate context, are not going to be valuable for this purpose. So this is the first thing I want to do. Then the next thing I want to do is I see if I can extract a transcript ID for each of these. And strictly speaking, yes, I want to do that. I want to only need to retrieve the record for a particular transcript ID. So we could do one of two things. We can either store the entire Gtf file into a data structure, which I think I'm going to do that way, but I could have also included the transcript symbol here and only got that transcript symbol, but for simplicity, this would be more educationally relevant to just read in the entire Gtf file in one fell swoop, and so that's not going to work. And if I paste, do I have that? Yeah, so I still save that. The transcript symbol, so this gives me an idea of what I'm looking for. So what I want to do is I want to see if transcript symbol is in the line. Maybe I'll do that. So I'll just say if transcript symbol in ATTR. And essentially say, if it's not in the ATTR, then there's probably an error. So if there is in the ATTR, I want to parse it out. And I'm going to need to use regular expressions to do that. But first, I need to import regular expressions. So I'm going to go ahead and do that. Jump to the beginning, and I'll just import it along with my RE here. And I'll go back to Gtf file. And what I want to do is I want to extract that out. So I'll say match equals re.search. And there should only be one instance of this pattern. That's going to be a pattern here and comma ATTR. And I really only need to search an ATTR. It's going to be more efficient to do that because I know it's going to be in that particular column of the Gtf file. And the pattern that I'm looking for is going to be this. And I just pasted it back in. And I want to be careful here. And these quotes, I think, are fine. What I want to do is I want to remove this, and I want to replace it with a placeholder of what I'm trying to capture. And so what's interesting here is that I can actually capture a particular portion of this pattern. And in order to do that, I put a pair of parentheses inside the quotes of what I'm trying to capture. And I need to put a placeholder of, or not really a placeholder, but a sequence of symbols here to indicate what I'm looking for. And I'm looking for anything. I'm really looking for any type of character, because if you see those gene symbols, they can have letters, numbers, dashes, whatever. So just a bit of an abundance of caution, I'm going to put dot star. So anything in quotes after the word transcript underscore symbol. And furthermore, I might want to do this with a question mark. This is a very standard regular expression that I like to use a lot. And so just to break it down again, what it means is the dot means anything. That's your wildcard character. It's going to match anything in the regular expression. And secondly, the asterisk says it can be any number of characters. It doesn't matter how long it is. Transcript symbol is going to be variable length, so I don't want to specify any length. The question mark says do this in a non-greedy way. The default behavior for regular expression is to find the largest match that satisfies these conditions, transcripts underscore symbol, space, and then in quotes, and then something in the quotes. And that could theoretically extend all the way to the end of the attribute column if the transcript symbol wasn't at the end of the column, and get everything to the next quote, including quotes in between. So I want to do this in a non-greedy way. I want to get the shortest match to this pattern, and that's going to be my transcript symbol. So then the transcript symbol itself will be stored in the group column. And so what I want to do is I want to say transcript symbol equals match.group. And I think I just want to put a 1 there to say that I want the first set of parentheses. You can actually use a regular expression to capture multiple patterns within these parentheses, and so everything that's in that's basically represented by the parentheses are going to be stored into this new variable transcript symbol. And so it's going to search through, find those records, parse them out of the quotes, and then and use that. So the other thing that I want to do is I want to put an else, because if I ever encounter an exon that doesn't have a proper transcript symbol, I have a problem. So I'm going to put print error parsing GTF. And people have told me, you know what, sys.exit, you actually don't need to use sys.exit, you can just use just exit. And there's also quit can be used here. That's fine, it's totally fine. You don't have to use sys.exit. I was using it because I was already using sys to read in the input arguments, but exit is perfectly fine here as well. And let's see, other things that I want to consider here. I want to, at this point, if I have this transcript symbol and I've properly parsed out the transcript symbol, I want to store information of this exon. So I want to now store for my data, I want to use the transcript symbol as my key, and I want to append to a list of tuples corresponding to the start, end, and strand. Let's just do that. So I'm going to put, and actually we're going to need the Chrome start, end, and strand. So let's do that. So I'm going to put append, and I'm going to store a tuple, and the tuple is going to be the Chrome, and then the start, end, and then strand. And I want to convert my start and end to ints. I want to go ahead and do that once and for all at the beginning here, because it's better to make those modifications at the very beginning as you're reading in the data, as opposed to sprinkling int variable data type conversions throughout your code. You want to do that once and for all at the beginning so that you can rest assured that it's going to be applied throughout the rest of the script. So at this point, I've now stored that, but the problem that I'm going to encounter, I know, is that every time I encounter a new transcript symbol, it's going to say, well, wait a minute, what are you appending to? Because data is just a dictionary. We haven't defined it as a list or a dictionary of lists. So what I really want to do is, if I encounter a transcript symbol that is not in my dictionary, I want to make the value of the dictionary with that as a key as a list. So therefore, I need to say if transcript symbol not in dictionary, which is called data, under that condition, I want to make data of transcript symbol equal to an empty list. And you can either put the square brackets or the word list, both should suffice here. And so now I have a situation where it's going to go through and check to see if there's a transcript symbol in the exon lines. If it is, I'm going to append this Chrome start end strand to a dictionary of lists, where the key is the transcript symbol. It seems like a pretty useful construct that we've got going on here. Anything else you want to modify? The one thing that I might modify here is to go ahead and convert to Python coordinates, meaning zero-based, non-inclusive end coordinates. And the way I would do that, starting off with the GTF, it's one-based. So what I want to do is I want to do -1 here for the start, because if the position is a particular position, like 736, in one-base coordinates, the corresponding zero-base coordinates would be over by 1, 735. Just subtract 1 from that number, and you should get the zero-based position. Now, the end position, I'm actually going to leave unchanged, because think about that. I could subtract 1 from it and get the zero-based position, but then I want to add that 1 back because it's non-inclusive. So I can actually leave the end position unchanged. And with that, I have a fully fledged transcript model. And what I might want to do is I might want to say if transcript symbol, and this is the symbol that was input from the user in transcript models, I want to print out the transcript sequence. So how about I do that? I'll just say print. I'm going to print the transcript symbol, and I'm going to print it just like a Fasta file. And then what I want to do is I want to loop through and concatenate the exons for this particular transcript symbol after retrieving them from the genome data structure dictionary here that we've created. So what I want to do is I want to say for exon in transcript models of transcript symbol, colon, and then I can unpack my exon tuples. So that's going to be a Chrome or seq name, start, end, strand. And then what I would want to do is I want to get the substring for that exon. So let's just say exon seq. That's going to be my actual sequence of the exon, or exon DNA, say, the DNA sequence of that exon. I want to do something like this. I want to do a substring. So I want to do a substring, but I want to do a substring of this particular chromosome here. So how would I do that? Well, recall that the genome is stored into this dictionary of strings here. So we did that in such a way that genome of Chrome would store the entire chromosome for that particular sequence name or chromosome name. And so this would be assigning exon DNA the entire chromosome. We don't want to do that. We want a substring. So if this is the entire chromosome sequence, in order to get a substring, recall, we apply bracket, bracket, and put start, colon, end, something like that. Now, the interesting thing here is this looks weird because we have a dictionary, so we have one set of square brackets because to specify the keys of the dictionary, and we have another set of square brackets to specify the substring, but it also kind of makes some sense. It makes a lot of sense, actually, when you look at it. And Chrome, start, end. The thing that's missing is the strand. So really what we want to do is let's assume that the strand is plus. So by default, it will be plus, and exon DNA will be initialized to be the forward strand of the DNA sequence at that particular start and end of that chromosome. But I can say if strand equals single, and put a single quote, whoops, and put a minus sign there because that's indicates on the negative strand. If that's true, then I want to do something like this. Exon DNA equals reverse complement of exon DNA. And furthermore, I can also put something like a variable here called transcript. I'll just call it transcript, and it's going to be an empty string. And I want to append to it these exons. But wait a minute. Before I do this, I want to be careful here. I could do it this way, but I'm not going to do it that way. What I'm going to do is I'm going to append to my transcript. I'm going to do transcript plus equals exon DNA. Let me go back to my Genome browser here. So if this is in the reverse strand, it's going to append these in reverse order, or in other words, it's forward order in terms of genomic position, but it's reverse order in terms of the ordering of the exons. I was actually transcribed in the reverse direction. So if I concatenate them in order and then reverse complement the whole thing, it should work. So the problem with that is the strand information is stored for each exon. So how do I do that? Maybe what I'll do is I could do the other way that I was saying, or I could just simply have a variable transcript strand, and I'm going to initialize it to be plus. I'm going to assume that it's plus, and I'm just going to update it as I go. Transcript strand equals strand. And this is the exon strand. And so, in other words, after I've looped through all the exons, transcript strand should store the strand of the exons. But actually, if there was a gene out there that had different exons that were different strands, this wouldn't work. But I'm assuming that's not the case. I don't know of any example like that. There may be weird examples like that, actually. But I don't know of any example like that, so I'm going to assume that all exons are the same strand. And this is, one of those classic assumptions that gets you in trouble, because anything you can imagine has probably happened somewhere in biology. But I'm going to go ahead and make that assumption here for now. And at the end, I want to say if transcript strand equals minus, then I want to say that the transcript is equal to the reverse complement of the transcript. And this should basically be the script. And in the end, I want to print it out. I want to print out the transcript. Now, barring any unforeseen complications, this should work. It very well may have errors, so let's just try and run it. So I'm going to clear my screen and Python extract transcript, and we've got an error already. So simply, I forgot my double equals. Oof. So seek type equals exon. Classic error, but I guess everyone makes it every now and again. So in other words, I was actually set using just single equals, which means assign the value to a variable, and that's not what I wanted at all. I wasn't trying to overwrite what seek type is to the exon each time. I wanted to check if it's equal to exon. So that's classic error, but easily checked in this case, I guess. So DM6 and then fly-based Gtf file. And then my transcript ID was CNC-RC. So let's see how this runs, if it does run. And we've got an error already. Gtf files not defined, so I simply go back and see how I define this. Okay, I did Gtf. Well, you know what? That's a different part of the code, so I'm not really sure what happened there. But I'm going to just go ahead and use underscores anyway to be consistent. And I didn't update this, just a big Gtf file. Try now. And there it is. There it is. That is the transcript of, that's it. So you know what? We can actually highlight this transcript in all its detail. And I noticed another error. This just prints out the word transcript symbol as opposed to the actual transcript symbol. I can fix that and see what happened there. But while that's running, I'm going to simply go here and open up Ncbi Blast. And I want to basically run blast n and see if I can find that this is in fact the right transcript. I'm going to blast this. Let's just go ahead and blast. And to nuke to NR. So that's going to hopefully find that nucleotide. And while that's running, I'm going to go back to my terminal. And apparently there's another error. I'm printing out the word transcript symbol as opposed to the actual transcript symbol, and that's because I have it in quotes. If you put something in quotes, it will do that. I want to concatenate the greater than sign with the transcript symbol. So check this out. If I did this, this is not perfect either, and I'll show you why. So it's running. Scroll back up. Look at that. It put a space. Now, if you recall, When I talked about Fasta files, there should not be a space in between the greater than sign and the ID. So the way we get around that is with string concatenation. By default, when you print with commas like this, it's going to put a space in between them. We don't want that in this case. We want the transcript symbol to be immediately adjacent to that greater than sign. So let's try one more time. And lo and behold, there it is. This is a perfectly formed Fasta file. If I were to redirect this into a my Fasta or something, that would be a perfectly formed Fasta file that would work. So let's check on that BLAST result and see how that worked. And here it is, Drosophila melanogaster chromosome 3R. Well, okay, yeah, it's going to blast to 3R, but more precisely, the cap and collar gene. And as you can see here, it is a perfect alignment, not a single gap. So therefore, I must have parsed those beginning and ends of those exons properly with my conversion to Pythonic zero-based non-inclusive end positions. And with that, I think we've done our task and we've successfully extracted the transcript sequence, taking into account both a multi-exonic structure of the transcript gene model, as well as taking into account the strand of the gene. And so with that, I'll close this video and I'll see you next time.